Good morning, everybody. Hope you guys are doing well. I am here to talk to you about the scientific method. So this is another topic that we're going to be moving into. We have finished with ecology with our last video, so that's pretty awesome. We are done with our first unit, and we are now moving on to our second. And the first thing we're going to talk about is steps to the scientific method. So the scientific method involves a series of steps that are used to investigate a natural occurrence. Right, so whenever a scientist, an investigator, wants to research and investigate something that's happening, they would use the scientific method to plan their research. Okay, so we're going to take a quick look at the steps that are needed to plan researching something and to plan experimentation before we actually go ahead and get started with planning our own. Right, just so you know what you're going to be doing. So the scientific method has a bunch of different steps. First being pose a problem or a question. You'd also then observe and research. You formulate a hypothesis, design your experiment, collect and analyze results, draw a conclusion, and communicate those results. So we're going to talk about all of those in a moment. So the first thing that you're going to want to do when designing an experiment is pose a problem or a question, right? You want to develop a question or problem that can be solved through experimentation, all right? So you have to first think of a problem or a question. What is something that you want to research? What is something that you are interested in learning more about? Something that can be solved through experimentation. So you can't just pick something that can't be proven or can't be solved, because there's not going to be really a path to experimentation with one of those types of questions. You want to be able to pick something that you're interested in, but that you also can figure out, right, through clear experimentation. That's always your first step in the scientific method. You can't design an experiment if you don't know what you want to research. So figure that out first. Your second step is going to be observation and your research. So make observations. So if you are interested in learning about how plants grow, right? How plants grow under red light. So that's your question. You are curious how plants grow under red light. You've now formulated your question. Now you're going to do your observation and research, as we see here. Look at plants, put plants under red light, see what happens, take notes, right? Write down some information about how those plants are growing, just based off of your observation, based off of what you see and what you've researched. Maybe look up different research papers for people who've done this, right? So observe, look up research from others, right? These are things that you're going to do to gain information about your topic before you actually start experimenting on it. Okay? You're then going to formulate a hypothesis. So once you figure out what you want to do, once you gather some information based on observation, based on research, you're then going to formulate a hypothesis. This is a very important part of any experiment, and you absolutely need to know how to formulate one and what they should sound like. So a hypothesis is a prediction to a problem. So you are predicting a possible answer to the problem or question. What do you think is actually going to happen? You don't know that this is going to happen because guess what? You haven't run your experiment yet. But that's not the point of a hypothesis. The point of a hypothesis is to make a prediction. You want to say to yourself, I believe based off of my observations that this is going to happen. And then when you do your actual experiment, you'll see if you were correct. So, for example, let's say that we want to do this experiment. If soil temperature rises, then plant growth will increase. This is a possible hypothesis to a research problem about temperature and plant growth. Does temperature affect plant growth? That's your question. Does temperature, right? Question is, does temperature affect plant growth? So you've now made your question, you've done your observations, and you've come up with this hypothesis that you believe if soil temperature rises, plant growth will increase. That's your hypothesis. You believe that. Now, a hypothesis, again, it's an educated guess about the relationship between the independent and dependent variables. We're going to learn what those two are in a second. When you create a hypothesis, I want you to always use an if-then statement. So we saw before, if soil temperature rises, then plant growth will increase. Before, when I mentioned my experiment about do plants grow better in red light, my hypothesis could be if plants are placed under red light, then they will grow more. I could also hypothesize if plants are placed under red light, then they will grow less. Right? It doesn't always have to be more. You could also go the opposite way and say that red light's actually going to be worse for the plants. So you can go in either direction. But remember, your hypothesis is just a guess. It's a prediction to what you think is going to happen in your experiment. Use an if-then statement to make a hypothesis very clear. All right? Your experiment could be about anything. Your experiment could be about, let's say your question is, do Jordans make you play basketball better? Right? That's your question. Do Jordans make you play basketball better? You could also say, if you don't like Jordans and you're a Nike fan, like I am, you could say, do Nikes make you play basketball better? Right? And that could be your question. Your hypothesis could then be, if you wear Jordans, then you will play basketball better. Or if you wear Jordans, you will play basketball worse. Then you will play basketball worse, right? So do you see how your if-then statement makes it easy to make a hypothesis, because you can kind of apply that to any type of question, anything that you want to experiment. All right? Hypothesis, very important. Now, we mentioned before that the hypothesis is a prediction based on the relationship between independent and dependent variables. Let's talk about what that is. An independent variable is the variable in your experiment that is manipulated or changed. So it's the factor that is intentionally changed by you, the experimenter. It is intentionally varied, right? It is manipulated. It gets changed. It's this factor that's changed during your experiment. And it's really important that you're able to identify independent and dependent variables. Those questions will come up all the time. So, for example, you want to test how different color bulbs affect the growth of a plant. That's your experiment. You want to test how different color bulbs affect the growth of a plant. So what do you think your independent variable is in this scenario? I think you should either pause the video or think in your head, what do you think the independent variable is? If you said the color of the bulb, you are correct, because that's what's changing. You're going to be using red bulbs, green bulbs, blue, orange, white, black, different color bulbs. And that is something that you are manipulating. You have the option to choose your color bulbs. You are picking the colors. That is changed in the experiment. You could also change it if you do the experiment again. Maybe this time you did red, blue, and green bulbs, and then you do an experiment with yellow, black, and orange bulbs. Right? That's changed. So your independent variable is any variable in your experiment that's actually manipulated or changed by the experimenter. Okay? Now, what's the dependent? Well, if the independent is the one that's changed, the dependent variable is the variable that responds to the independent. It is the factor that may change as a result of the independent variable. The dependent variable is dependent on the independent. And that's a lot of types of dependence, right? You're hearing that word often. But just remember, dependent variable sounds just like what it is. It's dependent on the independent. So the independent changes. We've manipulated that variable. That was the color bulbs. So in the experiment detailed before, what's the dependent variable? Remember, our question here was you want to test how different color bulbs affect the growth of a plant. And we've determined that the independent is the color of the bulb. Well, what is the color of the bulb going to do? What's dependent on the color of the bulb? Hopefully you said the growth of the plant. That's what we're testing, right? The growth of the plant. The plant growth depends on the color of the bulb, which is the independent variable. So in an experiment, the thing that is actually being measured, right? The thing that's actually being measured, okay? That is going to be your dependent variable. The thing that is being measured, right? Measured is your dependent variable. So remember, dependent versus independent, okay? Independent is what is manipulated. Independent is what is changed. Dependent is what is dependent on that independent variable. So what are we measuring? What is the result of our experiment? The growth of the plant. Very important that we are able to identify independent versus dependent. Okay? So after you figure out what your dependent and independent are, which you're going to have to do, right? You're going to have to actually state that in your write-up. Here's my experiment. Here's what my independent will be. Here's what my dependent will be. Then you can actually design an experiment. Develop and follow a procedure. So you're going to actually write out a procedure. What do you believe you're going to do to accomplish your results? How are you going to get results? What are you going to do? All right? So in the experiment before about the bulbs and the plant growth, my procedure will be detailed, but in short, it'll say something like, I'm going to gather 5 different plants. I'm going to place them in a row underneath 5 different color bulbs. I'm going to watch them grow over the course of a month. I'm going to record data every hour to see how much growth. I'm going to measure it in centimeters. I'm going to record that on a chart. You see what I mean? You're going to list out everything you do so that me reading it or anyone else reading it will understand how you did your experiment and how you got your results. You're going to write out your whole procedure. You're going to include materials list. You're going to say, I needed to buy, 5 different plants. What kind of plant was it? I needed specific pots. How many pots were there? What were the size of the pots? What type of soil was there? I needed, specific lamps to put the bulbs in. I needed an extension cord to plug it all in. You're going to write your materials. That's included in a write-up. And then the outcomes must be measurable. So you're going to actually talk about, what you expect to happen. Okay? Now, we developed a procedure. We listed our materials. And then we're going to determine our control group. So our control group in a scientific method is the group that serves as the standard of comparison. You always need to have a standard of comparison between your experiment and your control. So the control group is also called a no treatment group. And what that means is this group doesn't get experimented on. Okay? It's a control. So if you have 3 different plants that are all being tested under colored light bulbs, right? You have 1 plant under red, 1 plant under green, 1 plant under blue. Your control group would be a plant that isn't under any light. It's not being experimented on. There is no treatment. You're not applying the independent variable to this group. And we do that so that you have something to compare it to. Because you need to know that your results are a result of what you're actually testing. How do you know if the plants grew differently because of the light if you don't have a plant that wasn't under the light? You need to have some sort of comparison. So remember, a control group is a group that has no treatment. It doesn't get experimented on. It's just there to compare results with the groups that got experimented on. Okay? So you see here, the control group is exposed to the same conditions as the experimenter, except for the variable. Just like I said, the independent variable is not placed on this plant. So if you have plant 1, you have plant 2, you have plant 3, right? This one is under red light. This one is under blue light. This one is under green light. You will then have a fourth plant over here, okay, that is under all the same conditions. It's in the same type of pot. It's in the same type of soil. It's under the same type of temperature. Everything is the same, except there is no light above this plant's head, right? Notice how I didn't put a color up there, because we are testing color. That is our experiment, to see if color affects plant growth. But since this is our control, right? This is our control. There is no color placed above it, because we want to compare the colored groups over here to the one that isn't getting any colored light. That's our control group. Okay? We're going to make sure we have constants in our experiment. So constants are simply everything else that we just spoke about that stays the same. So these are all factors, except for the one being tested, that are kept the same. So you want to make sure all the plants are in the same type of soil, all the plants are in the same pot size, all the plants are under the same temperature, all the plants are in the same location, right? All the plants are being grown for the same amount of time. All of these things are kept the same so that your results are attributed only to the thing you are testing, right? Only attributed to the variable. So you want to keep everything else constant so that you know your results are actually based off of what you're testing and not off of something else, right? If you put your plants under different colored light, but you give one of them 10 times more water than the other, then you're going to see a growth in that plant regardless, because you're giving it more water. But that's not what you want to test. So you want to keep the water amount the same. That's a constant as well. All right, constants are very important. All right, so just another definition of constants. Okay, we got a couple of more slides. Trials. This is simply you repeating your experiment multiple times. Trials are necessary. Results are not always accurate, and you may get different results the second time around. You always need multiple trials to have something considered accurate. You can't just run one experiment and then say, all right, those are my results. Those are always my results forever and always. You can't do that. You got to run multiple trials to make sure you are accurate. All right. You're then going to collect and analyze your results. You may need to modify your procedure if you find something was wrong. You're going to confirm the results again by retesting. These are your trials. Okay. And you're going to include any tables, graphs, and photographs that you might need, right? So if you were actually running your experiment and you were getting data, you're going to put them in tables, you're going to put them in graphs, you're going to give pictures, maybe show us how the plants looked at the beginning, show us how they looked a week later through pictures, show us how the plants looked a month later, right? And then you're going to put in the graph and the tables your measurements that you got every single day, right? So you want to just make sure that everything is very clear. Right? You're then going to draw your conclusion. So you're going to include a statement that either accepts or rejects your original hypothesis. So if you ran your experiment and you saw that your original hypothesis was true, then you'll say, my hypothesis was accepted due to the data that I got, and you'll explain that. You might end up not proving yourself right. Your hypothesis could be wrong, right? You're going to reject it then. You're going to say, well, my original hypothesis was that plants would grow best under red light. That was rejected because I didn't see that in my data. All right, so you'll have to state that in your conclusion. And then you'll also make recommendations for further study and possible improvements. What do you think you could do next time? Or what could I do if I wanted to run this experiment? How could I make it better? What do you think I could do? You're going to communicate your results, right? So be prepared to present to an audience, expect questions. You're going to tell everybody what you did. You gotta be able to present those, especially if you're doing a real scientific experiment that could get published, right? People will read it. People will know about it. Make sure you understand your data and you understand your experiment so that you can communicate it to other people. And that is it. All right. So we're going to go through this. Make sure you take notes on it. Ask any questions if you have any, because tomorrow you're going to design your own experiment in class using everything that we just went through today. All right, guys. Have a great rest of your day. We'll talk to you later.